Hey guys, welcome. This is another trip through Genbank and also ExPasy, which is this. This is the Bioinformatics Resource Portal. And right away you can see a serious amount of tools available. This is kind of where we're going with our first inquiry, which is upcoming. I just hadn't posted it yet. Today's Tuesday afternoon, and let's go. So here we go to Genbank overview. It's at the National Library of Medicine, nih.gov. Here we go, Genbank. So right away, there's a lot of information thrown in to different ways to deal with that. You can submit data, you can search it, you can update the records, lots of information here. You'll need to spend some time exploring. What I'm going to do is go to protein, and this is one of my favorite proteins, Rab11, and this is from my organism, Dictyostelium. So I hit search, and it's hunting for the protein page for this particular protein in my system. Here it is. This is the accession. It's got a number here, Unipore KB Swiss protein. There's actual identification number. There's a lot of things we can do at this point. Here's over here, analyze this sequence. We could hit blast. So we could find regions of similarity between this sequence and other sequence using the blast. We can identify conserved domains. We can highlight those and find in the sequence. Here are some other computationally incurred pathways, translational modifications, similar protein sequences. We can look up papers, the taxonomy of the slime mold, lots of information here, and you can see it goes all the way down. And this is a lot of information. This is RB11A DIC Dictyostelium accession number. So here's another number that you could go and look at, and this is the protein number. So if I hit accession number here, it should pull up the protein. Here it is at the uniport, and there it is, RAS related protein Rab11A and Dictyostelium slime mold. And right away you can see they've isolated domains, or what they call key features, and this is amino acid 20 to 27, 68 to 72, and 126 to 129. The molecular function of Rab11A is a GTPase protein, has GTPase activity. It will bind GTP, and it interacts with other proteins that are involved in exchanging and activating it. These are some of the functions that Rab11 has been found to work in, and notice some of the sources like contractile vacuole organization. So Rab11A actually is involved in my organism, and so when we go there, this is gene ontology, and let's see what this is doing for us. Biological process, it actually defines that process, and it defines what a contractile vacuole is. And so I think, let's see, if I hit that, what do I get? Is that the, oh, not sure, I should have been there. Anyway, we'll jump out of that and we'll go back to the pages here. Okay, and we'll go back to where we were. But you can have a lot of fun, that's what I call fun, with looking at Genbank here. So if you'll go down the original accession page here, this is what we're looking at: keywords, source organism, authors. Who is this person here? Do we believe this paper here? Hey, J. Bush, that would be me, and this was the paper. And if you wanted to look up that paper, you could. See, okay. And we submitted the sequence, and these are some of the features of the sequence. So the gene, for instance, is 214 amino acids, and there are known regions. Okay, and there are any numbers or amino acids. And there is Rab's family motif 1 is 11 to 15. Then there's 20 to 27. So they define regions, at least their data. So if you go all the way down, this is the actual sequence here. So that's the actual sequence. And let's see, I may be able to do the FASTA, I don't know, yeah, FASTA. Let's hit FASTA. Should just give me the protein sequence. Yeah, so if you want to do further manipulations of your protein, you need to get it in the FASTA format. Notice the numbers went away, so it's just pretty much the one letter amino acid sequences. So I do Control C for copy, then I go here and I go to Swiss Model Worksite, and let's see here, start modeling. See, they're excited. Welcome to Swiss Model. Let's see what happens. I haven't done this, so paste your target sequence. So I'm going to paste. Okay, and let's see, build a model. Let's see if it lets me build a model. I may have had to put a project down or an email. Auto model is running. Okay, so it's, I think, modeling my protein here. Let's see what happens. I haven't done this before. Click model in 3D. So I probably need to get my ad block pause on this site. Okay, so I shut down my ad block maybe. I don't know. Yeah, okay. Now this may take a while. So the auto model is running. So while that's happening, let's go back here and I want to show you. Here's a button here, conserve domains. You hit this and it gives you a cartoon of your protein, and here it is, and here we go. So what happens, this is from 0 to 214 amino acids. These little blue triangles have a function. This is GTPA magnesium 2 binding sites, there, and it actually defines what that domain is. Okay, and then here's a GEF protein that interacts in this region. This is a GDI interaction at this region. It's where some handshakings. Effectors, these are other proteins, will bind in this region. This is the G1 box, the G2 box, or so-called switch region. And you can see here's motif 2 in this region. And way down here is another motif. So this is the amino acid, and these are key domain areas that you can access and study. And here's some papers on looking at these key domains as well. So I can zoom to the amino acids. So you can see, for instance, these bold amino acids. 1, 2, 3, 4, 5. Here they are. D, Y, L, Y, K are these 5. And that's involved in Rab's superfamily motif 1 function right there. Here are these amino acids. And there they are. That is binding GTP magnesium right there, those amino acids. So in molecular and cell, how would you prove that these are critical proteins, amino acids for function? You could mutate and get rid of that, and this protein would not be able to bind GTP. Just getting rid of these amino acids, or maybe even one of them, you could do one at a time and test binding. Okay, so this sets up work that you can do to test for functionality. So the in silico biologists give us this information based on homology throughout all the known proteins. And then we, if this protein's important enough, could go in and manipulate the protein and make versions, mutant versions of the proteins to study and determine areas of functionality here. So you can kind of follow these key amino acids here that are in bold, and they're showing the actual sequences and what's going on as you roll through here. And there's other options as well. So for your inquiry, I'm going to basically want you to go through. I'm going to assign each group a protein, and then maybe even any protein I hadn't decided yet, and let you guys generate the data. And what does it mean? So that means you're going to have to figure, well, what is motif 1? What is that? Well, I need some more information. So in your inquiry report, you'll get that. So we're going to get deeper into it, but this is just the easy first one, just so you understand Genbank and Swiss modeling, for instance. And it looks like it's still modeling. I don't know. This may take hours. I have no clue how long this modeling take, but I know there's a lot of information that can be generated. But I at least wanted to start you guys out on this, and we'll see where we end up a little bit. We'll try to work on this Thursday, but now you've seen the ExPasy and the Genbank a little bit. So maybe one day I'll get a model here. We don't have time to wait for this, but and again, this may be something you have to do elsewhere. But right now, that was just an introduction to these two areas. Maybe I should just go back here real quick. Okay, give up the modeling. We give up Swiss model. You win. Okay, template, user, deep view. See, I don't know what the colors, isn't that interesting? Target template alignment. Let's go back a little further here. No. I was hoping to get a protein model like that. Not necessarily a donut, but something here. Protein secondary structure. Okay, let's see. Wonder what's secondary structure. Let's see if it gives us an opportunity to do secondary structure. So looks like in FASTA format, notice you'll see in FASTA format predict. So this is secondary structure, and did it not like my FASTA format? Ooh, so I wonder if I did it wrong. Maybe let's try again. Did I not put it at the first? Ooh, it doesn't like that format, does it? I wonder if there's something about the cut. Oh, placed. Let's try paste as plain text. Let's trick it. Nope, didn't like that either. Don't know if it's broken. Well, that blanks, doesn't it? Yeah, it does. Hey, fine. We're through with you. We'll go find another site. See if I can find another site. Maybe it won't let me out. I'm stuck in protein hell. Let's see, what is this? Protein molecular modeling, domain linker prediction, protein homology, coiled regions. Let's see if this thing has any coils. I have no clue if it has coils. Where's our fast? Plain text. Okay, I can do that. Run coils. Oh, so it looks like there's some type of coiled right in these amino acids here. Okay, hey, I can dig that. Now, again, what is the function of this protein in this coiled region here? We could get rid of that and see what happens. So that's neat. We can get you that information, but you have to know what coils are a little bit and structure and what it might do. Let's see what other things. So you get the feeling here. Helical content. Let's see if I can predict the helical content. Let's see here. What are we doing? Predictive algorithm, news, links. Is not a program. 100 peptide, Tango. Is it predictive links? I wonder how. Jump into this website, please. Oh, the calculation is now log in, so there may be some login. Well, that blanks. So you might have to play. You're in your group, just see what all the data is. So that's part of it, is figuring out what beta turns. Okay, let's see. Generate movies of protein dynamics. Woo. Let's see. Here's our friend FASTA. Okay, and then I'm just going to hit submit it. See what happens. See if it blows up here. Processing. This may take a while. Boy, that's a hell of a job number, isn't it? Processing. Looks like I could get an email if you guys are finding a site. Sometimes they'll let you plug in your email here. So maybe, yeah, so it's going to send it to my email when it's finished. Yay. So I can get back out of there. Okay, well, I think you got the idea. We're just kind of using this inquiry to kind of tenderly bring you along. Okay, more of me talking later. Let me see if I can figure out how to end this. Control